Bienvenidos al podcast de la Sociedad Latinoamericana Interdisciplinaria de Cannabinología, SLIC. En el 2026, este podcast reúne a 5 investigadores de renombre internacional y pioneros en el estudio del cannabis, los cannabinoides y el sistema endocannabinoide. Sus contribuciones han sido fundamentales para dar forma al conocimiento científico actual en este campo. A lo largo de décadas de investigación, su trabajo ha definido conceptos clave, abierto nuevas vías terapéuticas y contribuido de manera decisiva a la historia de la ciencia cannabinoide. Mi nombre es Paola Cubillos, soy miembro fundador de SLIC y nos llena de orgullo poder ofrecer este espacio de diálogo y aprendizaje para la comunidad latinoamericana. Bienvenidos. Hoy tenemos el enorme placer de presentar a un referente indiscutido en la investigación en cannabinoides, el profesor Mauro Maccarrone. El profesor Maccarrone es catedrático de bioquímica de la Universidad de L'Aquila en Italia y jefe de la unidad de neuroquímica de lípidos del Centro Europeo de Investigación Cerebral en Roma. Su trayectoria científica es realmente excepcional. Ha publicado extensamente en revistas de alto impacto con más de 24.700 citas y es autor de 9 patentes, lo que reflejan la relevancia translacional de su trabajo. Ha sido una figura clave en el desarrollo del campo de los cannabinoides. Fue presidente de la Sociedad Internacional de Investigación en Cannabinoides, presidió la Gordon Research Conference sobre la función de los cannabinoides en el sistema nervioso central y ha recibido prestigiosos reconocimientos como el premio de la IACM y el premio Meshulam de ICRS. Sus contribuciones científicas, desde los mecanismos celulares de la anandamida y el papel del sistema endocannabinoide en la inflamación crónica, hasta la señalización del 2-AG en el cerebro y la completa relación entre cannabinoides y fertilidad, han marcado profundamente nuestra comprensión del sistema endocannabinoide y continúan influyendo a generaciones de investigadores en todo el mundo. Para nuestra sociedad científica es un verdadero honor contar con el profesor Mauro Maccarrone como miembro de nuestro Comité Honorario de Notables. Su apoyo y cercanía representan no solo el reconocimiento a nuestro trabajo, sino también una inspiración para seguir fortaleciendo la investigación rigurosa y colaborativa en nuestra región. Una nota para quienes nos escuchan: la conversación con el Dr. Maccarrone se llevó a cabo en inglés. Sin embargo, después de la intervención del ponente, incluiremos apartes traducidos en el español para facilitar la comprensión de quienes nos escuchan en Latinoamérica. De la misma manera, tendrán acceso a la transcripción del episodio tanto en inglés como en español en las notas de este episodio. Nuevamente, Dr. Maccarrone, un placer que esté con nosotros. So welcome, Professor Maccarrone. Thank you so much again for agreeing to join us in this podcast. I just mentioned in the introduction of the episode how grateful we are that you have agreed to be part of our honorary committee and for your continued support and your willingness to share your insights and part of your journey as a cannabinoid and endocannabinoid researcher with a Latin American audience. So thank you so much for being here with us today and being willing to share. So as one usually goes, asking people, who are prominent in the field, this is a field where you don't find a lot of people jumping into necessarily. It's slightly controversial, even less so nowadays that we've had so much science to back up many of the things that have been discovered with respect to the potential of cannabinoids. But this is not a field that one jumps into willingly. So there's usually a backstory to a lot of the folks that decide to jump into this field of research. What is your story? What brought you to the endocannabinoid field research field? Were there any specific moments or experiences that influenced that decision for you to join this exciting field?
Thank you so much, Dr. Cubillos, Paola, if I may say so. The pleasure of being part of this podcast series is really mine. So, to your question, actually, I did not really jump into the field because when I started investigating endocannabinoids, there was not yet a field. Back in 1995, the second endocannabinoid was discovered, 2-AG, by Rafi Meshulam and Sugura independently in Jerusalem and in Israel and in Japan. And for only 3 years, anandamide was known. And that is because of anandamide that I was engaged in this emerging area because I was studying very much in detail one enzyme, lipoxygenase, still very popular, one of the ones making eicosanoids. And it was very recent, the observation that anandamide could be a substrate of this enzyme. So because of the enzyme, I wanted to get the subset, and I was immediately attracted by the fact that with a little change of the molecule, an arachidonic acid with an extra piece becoming anandamide, new compounds could be generated by lipoxygenase and other entities. And we started to develop methods for measuring through radioactive stuff, receptors, metabolic enzymes, and since then we never stopped contributing to this field.
El profesor Mauro Maccarrone inicia agradeciendo a la doctora Cubillos y expresa su satisfacción por participar en la serie de podcast. Explica que no entró de manera intencionada en el campo de los endocannabinoides, ya que cuando empezó a investigarlos todavía no existía como tal ese campo de estudio. Relata que en 1995 se descubrió el segundo endocannabinoide, el 2-AG, por Rafael Mechulam y Sugiura de manera independiente en Israel y Japón, y que anandamida solo se conocía desde hacía 3 años. Precisamente, fue el descubrimiento de la anandamida lo que le llevó a interesarse por esta área emergente, ya que investigaba en profundidad la enzima lipoxigenasa, muy popular en aquel momento, y observó recientemente que la anandamida podía ser un sustrato de dicha enzima. A partir de ahí, le atrajo la idea de que, con un pequeño cambio molecular en el ácido araquidónico que da lugar a la anandamida, se podían generar nuevos compuestos gracias a la acción de la lipoxigenasa y otras entidades. Junto a su equipo, comenzaron a desarrollar métodos para medir receptores y enzimas metabólicas utilizando técnicas radioactivas, y desde entonces no han dejado de contribuir al avance de este campo científico.
I think it reflects also the experiences of other researchers in the field, that they start sort of discovering things anew, and then they realize that many of these findings are converging around the endocannabinoid system, which is something that definitely leaves a lot to be explored still. We don't know everything there is to know about this system. And you know, the usual discussion point for many of us in the field is that this is not even taught in medical school or in any of the other sciences in the professions. And unfortunately, that is the case because there's so much richness to this endocannabinoid system and to this ability to control so many things in our physiology. And I'm so glad that people like yourselves are contributing to our understanding and to further the discussion in this area. So now, you have done a lot of work in this field, exploring many different areas from the neuroinflammatory conditions, understanding the lipid metabolism that has to do with the endocannabinoid system, understanding perhaps the intersection between fertility and the endocannabinoid system as well. So among your many, many contributions to this field, which one do you think that has the most lasting impact, and why do you think that is?
First, let me say that when I started to be curious about endocannabinoids, this emerging area was actually a territory for pharmacologists. And as a biochemist, I was more curious about mechanisms of action, molecules that could play a role in the biological activities of these new compounds. And I had the great pleasure and luck, even luxury, I would say, to collaborate with so many colleagues, especially on the clinical side, neurologists rather than cardiologists and so on and so forth, which gave me the chance to explore kind of my biochemistry, the knowledge I had on the biochemistry of the compounds in so many different setups and areas. So actually quite a large array of contributions. But among these, I would say that within the central nervous system, one that was really, I think, strong has been to show that anandamide and 2-AG can regulate each other, can in a way crosstalk via enzymes, which at those times something around 2010, was not clear yet. The two endocannabinoids seem to be kind of sort of disposable, doing more or less the same things. But in fact, that was not the case. In the brain, one endocannabinoid could modulate the other, and this could have an impact on excitatory versus inhibitory neurotransmission. In the periphery, as you anticipated, maybe the most striking evidence was about reproductive events, and in particular human miscarriage. The fact that a single enzyme in blood could anticipate already at the 7th week of pregnancy that something could go wrong later on, much later on. So potentially becoming a biomarker of this spontaneous miscarriage that still now does not have a real biomarker or mechanism behind. So endocannabinoids and human reproduction, female reproduction, and then also we could give quite some contribution to male reproduction. I think that is the most unexpected and rewarding area where we could give a contribution.
El doctor Maccarrone relata que cuando comenzó a interesarse por los endocannabinoides, este campo era principalmente explorado por farmacólogos. Como bioquímico, su curiosidad lo llevó a investigar los mecanismos de acción y las moléculas involucradas en la actividad biológica de estos compuestos, lo que le permitió colaborar estrechamente con colegas del ámbito clínico, en especial neurólogos. Esta colaboración le brindó la oportunidad de aplicar sus conocimientos bioquímicos en diversos contextos, logrando una amplia gama de contribuciones. Maccarrone destaca que, en el sistema nervioso central, uno de sus aportes más importantes fue demostrar que los endocannabinoides anandamida y 2-AG pueden regularse mutuamente a través de enzimas, interactuando y modulando la neurotransmisión excitatoria e inhibitoria, lo que representó un avance novedoso en la época. Asimismo, en el ámbito periférico, resalta su investigación sobre eventos reproductivos. En particular, el hallazgo de que una enzima en la sangre podría anticipar desde la 7ª semana de gestación la posibilidad de un aborto espontáneo, lo que abre la puerta a identificar potenciales biomarcadores para este fenómeno, que hasta ahora carece de mecanismos claros. Finalmente, Maccarrone considera que los estudios sobre endocannabinoides en la reproducción humana, tanto femenina como masculina, han sido el área más inesperada y gratificante de su trabajo, permitiéndole realizar aportes significativos al conocimiento en este campo.
Excellent. And I imagine that it's not an easy area to research. There are so many restrictions in terms of the kinds of materials that one can get access to, and also potential, if one starts manipulating things, potential ethical questions that may arise as well with trying to understand how the endocannabinoid is regulating all this multiple, mechanisms that come into play to create an embryo or to be able to that embryo to progress further along in the reproductive or in the pregnancy cycle. So thank you for focusing on that, because there's not a lot of people who decide to jump in this really tricky and potentially controversial areas as well. And definitely fertility is an area that overall as a whole is understudied. And the more information we can get, obviously, the better decisions we're going to be making as, people who are interested in trying to understand what their reproductive abilities may be. So, which has been the most surprising finding in your research, or through any of the research of your colleagues, a finding that you say, we did not expect this at all, and that perhaps broke a paradigm that existed in the field, or that assisted us in looking at things in a different way?
Well, this is a difficult question, but overall, I think that the surprise, at least to me, was that there is one system, the immune system, that is behind so many others. In the central nervous system, when you think of neuroinflammation, for instance, or in the periphery, when you think of reproduction. Because in the new field of lipid signaling in reproductive events, what was really striking is how the immune system has an impact on that. So I did not expect that our sentinels, our army, could be really behind almost everything. and how by finely tuning this, the response of this immune system, you could end up with physiological responses, unfortunately likely to become pathological responses. And endocannabinoids are the type of signals that are perfect to that, because they are not so stable, and therefore if you use the amounts of endocannabinoids and their ability to trigger a receptor-mediated signal transduction pathway, the amount of endocannabinoids as the trigger, so that when you have too much or you have too little, you can bring that organ towards physiology or pathology, is so fascinating. There is nothing black or white. There is the right amount in the right place at the right time for everything.
La doctora Paola Cubillos plantea una pregunta sobre el hallazgo más sorprendente en la investigación del profesor Maccarrone o la de sus colegas, especialmente aquellos descubrimientos que rompieron paradigmas existentes o permitieron ver el campo desde una nueva perspectiva. En respuesta, el Dr. Maccarrone comparte que lo más inesperado para él fue la importancia central del sistema inmunológico en diversos procesos, desde la neuroinflamación en el sistema nervioso central hasta la reproducción. Destaca cómo, en el campo de la señalización lipídica relacionada con eventos reproductivos, el impacto del sistema inmunológico resulta ser realmente significativo. El profesor Maccarrone expresa su sorpresa al descubrir que nuestro ejército inmunológico está detrás de casi todo y que ajustar finamente sus respuestas puede generar resultados fisiológicos, aunque desafortunadamente también puede conducir a respuestas patológicas. En este contexto, los endocannabinoides juegan un papel crucial, ya que su estabilidad y cantidad pueden determinar si un órgano funciona correctamente o cae en la patología. Finalmente, el Dr. Maccarrone reflexiona sobre la complejidad de estos sistemas, señalando que no existen respuestas absolutas. Todo depende de encontrar el equilibrio adecuado en el momento y lugar precisos.
The right amount at the right place at the right time, which we have not found yet. At least not when it comes to, the conventional or the existing ways of trying to manipulate the endocannabinoid system into trying to find that balance, right? So exactly. It's a very tricky area. Again, the inflammatory system, as you posted, is something that we're just trying to understand the huge significance of for many, if not most, conditions that affect human beings. So again, it's another area that it's really worthwhile investing additional time and thinking into, which leads perfectly into my third question and my fourth question. I don't know how many I've asked already. Your work lies at the intersection of the exploration of the endocannabinoid system and finding new therapeutic avenues for neuroinflammatory diseases. What questions in this area have you found the most challenging or the most rewarding to answer?
Yeah, again, a very good question that also brings me to think of the future. If I have to say kind of plain, I think I would say that is the complexity of how endocannabinoids can cross-talk with other mediators. And while we were used for the last decade, more or less, to think of endocannabinoids as a pro-homeostatic regulators, able to have a higher place in the hierarchy of mediators, now we are learning that they do act as pro-homeostatic signals, but they do so by continuously confronting with other signals that are very often lipids. And this layer of complexity cannot be neglected, cannot be ignored, especially when you want to end up with something good for health. And tell you that it's not so surprising that it is difficult to end up with a good drug targeting the endocannabinoid system. It's not just a matter of switching on, switching off something, but again, is a matter of giving the right tone for that condition. And this is not so easy to do. And maybe we want to discuss it in more details. It's not only a matter of a drug able to selectively hit a certain target, but again, it has to hit it only in those cells that are engaged in that specific disease or that specific condition. You cannot do it everywhere, and everywhere does no longer mean in the central nervous system versus periphery. It's not enough that you have something that does not penetrate the blood-brain barrier. It means that within the immune cells, you may want to hit only plasmacytoid dendritic cells, but not myeloid dendritic cells. So dendritic cells, a subpopulation of immune cells, but that is not enough for the endocannabinoid regulation. One certain type of that specific subset of cells can be activated, but the other should not. So we confront with a very high level of complexity in these signals.
En respuesta a la pregunta sobre los retos y satisfacciones más significativos en el ámbito del sistema endocannabinoide y las enfermedades neuroinflamatorias, el Dr. Maccarrone expone que uno de los mayores desafíos consiste en comprender la complejidad de las interacciones entre los endocannabinoides y otros mediadores biológicos. Durante la última década, se ha considerado a los endocannabinoides como reguladores pro-homeostáticos, pero actualmente se reconoce que estos no actúan de forma aislada, sino que interactúan constantemente con otras señales, especialmente lípidos. Esta complejidad impide ignorar la importancia de estas interacciones, sobre todo cuando se busca el desarrollo de terapias eficaces para la salud. El profesor Maccarrone señala que diseñar fármacos dirigidos al sistema endocannabinoide no es simplemente cuestión de activar o desactivar una función, sino de ajustar el tono adecuado para cada condición, lo que resulta complicado. Además, destaca que no basta con que un medicamento seleccione un objetivo concreto, debe hacerlo únicamente en las células implicadas en la enfermedad o condición específica, lo que añade otro nivel de dificultad. Por ejemplo, dentro del sistema inmunitario, el tratamiento podría necesitar dirigirse solo a ciertas subpoblaciones de células dendríticas, pero no a todas, ya que la regulación por endocannabinoides es altamente específica y compleja. En definitiva, el Dr. Maccarrone enfatiza que enfrentarse a este alto grado de sofisticación en las señales celulares es uno de los retos más relevantes y estimulantes del campo.
That is such an excellent point that you're making, and I appreciate you making it because in this field, one, because there's so much that is still unknown, and we've only known the cannabis plant as it is, and the two main compounds that we understand better, although CBD, there's so much more that needs to be discovered with respect to CBD, but THC is a much simpler molecule to study, to analyze. Thanks to THC, we discovered the endocannabinoid system.
Right.
But there are many folks, many people who believe that you can actually modulate the endocannabinoid system through external cannabinoids, aka THC. So what would you say to those who still believe in this simplistic view that you can achieve such modulation when in reality you have painted a much more complex picture?
Yeah.
And not even in terms of the location, periphery, central nervous system, but the location of the endocannabinoid receptors in the different cells, in the different types of cells, in the sort of function that they exert. So could you speak a little bit about modulating the endocannabinoid system through THC mainly?
The phyto. Yeah, this is all, again, a very central question that I'm afraid I have to say the scientific community has underlooked for so many years. The simple point is the following. Phytocannabinoids cannot be metabolized by our cells. We, by a flick of nature, have receptors, very important receptors, where phytocannabinoids can be active, although the chemical structure of phytocannabinoids like THC or CBD is totally different from the structure of endocannabinoids. One is a terpenophenol compound that mammalian cells, animal cells will never be able to build. Our compounds are little derivatives of a fatty acid like arachidonic acid, mainly. So different chemical structures that by accident in the three-dimensional space expose 3 points that are equally well to bind the receptors. So phytocannabinoids are active in our body, but unlike the endocannabinoids, they cannot be metabolized. nor synthesized, as I said, but maybe more importantly, not degraded. They cannot be hydrolyzed. So if you take what I said before, that endogenous cannabinoids work with a very tight regulation of the amounts and the ability then to trigger receptor-mediated, maybe a number of non-receptor-mediated effects. And now you add to our body something that cannot be regulated because we cannot do anything with it until we excrete it. And we do that by conjugating it with something else, and then we eliminate it in the urine or in the feces. If you again now compare an endogenous molecule that is subjected to an absolutely tight metabolic control and another that escapes it totally, you immediately appreciate that what we can expect from an endocannabinoid signaling cannot be expected for a cannabinoid signaling or phytocannabinoid signaling. Which means that again, when we want to use phytocannabinoids for medicinal purposes, we have to be very, very careful in defining what are we using, what are we giving to a patient. And this seems something obvious with the tools we have today, but it's not done. We never standardized the plant extract, the formulations used as therapeutic compounds, for instance. And to me, this is a very big problem because we don't learn out of the effects of a plant extract because you never know what was behind that plant extract. I mean that not only knowing the amount of THC or now of CBD is enough to say what that extract can do. There are so many other compounds, phytocannabinoids, more than 110 different compounds belonging to 10 different families. And on top of that, more than 400 non-cannabinoid compounds in a cannabis extract, that until we appreciate what these molecules, this ensemble of molecules can do in our body, we will not know the real consequences and if they can modulate the endogenous system or simply disturb it heavily, like something that breaks a very sophisticated engine, blocking the wheels to move one or the other, if I can make the point. So we have to be extremely careful.
En esta parte de la conversación se aborda la complejidad de los receptores endocannabinoides, destacando que su ubicación y función varían según el tipo de célula. La doctora Cubillos le pide al profesor Maccarrone que explique cómo se puede modular el sistema endocannabinoide a través de compuestos externos, principalmente el THC. El doctor Maccarrone menciona que, durante años, la comunidad científica ha pasado por alto la diferencia esencial entre los fitocannabinoides, como el THC y el CBD, y los endocannabinoides producidos naturalmente por el cuerpo. Aunque los fitocannabinoides pueden activar receptores importantes en el organismo, sus estructuras químicas son muy distintas a las de los endocannabinoides, y además, el cuerpo no puede metabolizarlos, sintetizarlos ni degradarlos, como ocurre con los compuestos endógenos. Explica que los endocannabinoides están regulados de manera muy estricta dentro del organismo, mientras que los fitocannabinoides escapan a este control y solo pueden eliminarse una vez que el cuerpo los excreta. Por lo tanto, no se puede esperar que la señalización de los fitocannabinoides produzca los mismos efectos que la de los endocannabinoides. El profesor Maccarrone enfatiza la importancia de ser cautelosos al utilizar fitocannabinoides con fines medicinales, ya que no se han estandarizado los extractos ni las formulaciones de la planta, lo que representa un gran problema para entender sus efectos reales. Además, subraya que conocer únicamente la cantidad de THC o cannabidiol no es suficiente, ya que existen muchos otros compuestos en el cannabis que pueden influir en el sistema endocannabinoide. Hasta que se comprenda la interacción de todas estas moléculas en el cuerpo, no se podrá saber si realmente modulan el sistema endógeno o si, por el contrario, lo alteran de manera negativa. Por ello, concluye que se debe tener extrema precaución en su uso.
That is a great way that you phrase the necessity or the need to carry out research at different levels as way to understand the short-term impact on the cellular, the physiological, the systems level, and in the long term as well, in terms of not only symptom management or symptom control, but also the long-term impacts of long-term exposure to cannabinoids. The research that we have so far is just very low quality, a lot of observational research, retrospective studies that speak more to either the benefits that people report and also the adverse events that people report. But going prospectively and trying to fine-tune our ability to ascertain exactly what's going on, it's something that is definitely missing. That's why sometimes as clinicians, we have to make a lot of decisions with respect to cannabis medicines in the context of uncertainty, because we don't really know.
Right.
And I hope that yeah, go ahead.
Yeah, and Paola, let me add one point that is a kind of optimistic view anyway. It is clear that there is a lot of potential in phytocannabinoids as therapeutics. And one example is a very nice study, although preclinical study, by a German group led by my colleague and friend Andreas Zimmer, showing how THC in young animals can do a disaster in learning, memory, and so on and so forth. While in old animals, it can be beneficial. And you know why? Because the background endocannabinoid tone that you find in young animals, where the tone is very high, and in old animals, where the tone is going down, is so different. Just to say that even the same dose of a phytocannabinoid given to two different persons, because one has a high level, a very active endogenous system, and the other has some defect somewhere, can lead to opposite effects. And now we can know these things. We could profile the endocannabinoid system of each of us, maybe with a few drops of blood. And would be wise to try to do this if then we want to use phytocannabinoids as therapeutics.
El profesor Maccarrone menciona un estudio preclínico realizado por un grupo alemán, liderado por Andreas Zimmer, que demuestra cómo el THC puede causar graves problemas de aprendizaje y memoria en animales jóvenes. Sin embargo, en animales mayores, este mismo compuesto puede resultar beneficioso. Esta diferencia se debe al tono del sistema endocannabinoide. Mientras que en los jóvenes es muy alto, en los mayores disminuye. El Dr. Maccarrone subraya que incluso una dosis idéntica de un fitocannabinoide puede producir efectos opuestos en distintas personas, dependiendo de la actividad de su sistema endógeno. Por ello, sugiere que sería prudente analizar el sistema endocannabinoide individual, por ejemplo mediante un simple análisis de sangre, antes de utilizar fitocannabinoides con fines terapéuticos.
So I think you're talking about potentially moving more into the pharmacogenomics kinds of discoveries that one can sort of be able to understand the impact for the individual depending on the types of compounds that are being used, and understand whether they're beneficial or it could be detrimental to their health. So that is definitely really interesting. Would be a really interesting area of research to continue furthering and moving towards. But yeah, but for now, I think as clinicians, the lack of good quality studies, at least to support the short-term symptom management, that the randomized controlled trials are really limited in that way, that they're very short-term and they're just really limited. They focus on an individual group of people. So the evidence is definitely not something that exists for one to make confident decisions. And what you mentioned with respect to this potential impacts on a systems-wide level is something that we definitely do not have a good grasp on, but hopefully we'll be able to understand more as time goes on, as more people sort of jump into this field and start making more discoveries. Now, looking into the future, and because since you already expressed some areas where you think the future will go in terms of this field, what is an unresolved debate or an unresolved question that you have found in this field that you consider to be particularly important? You talked about understanding at the cellular level what happens with the phytocannabinoids when they're used. Is there any other area that you, or a question, research question that you think that the field should be focusing on?
Well, yeah, I think that here we could have a chapter that is endocannabinoid transport and maybe also phytocannabinoid transport. So, we have an entity that was discovered through drugs very early, the so-called anandamide membrane transporter, at those times meaning only a mechanism whereby endocannabinoids can cross the plasma membrane, so getting in and out the cell. After so many years, this entity remains vague. We don't know if it exists, if it can be in some cells but not in others, if it is a single protein or whatever. I think that it would be important to solve this issue, maybe finding out that there is not a single protein, but an ensemble of proteins that have to go together to form this transporter, whatever it is, a carrier, a channel, whatever. And that is why you don't have a single gene. It's not easy to get the structure out. But it is important because drugs show that by manipulating the transport mechanism, you can have therapeutic effects. More in general, and again linked to your question, I think that, and this can be a lesson for the younger scientists, we know too little about the transport in general, meaning how endocannabinoids move in the cell, in the cytosol, in an aqueous milieu, and how they do outside the cell. Just think of the synapse, and you have these compounds moving from the postsynaptic to the presynaptic neuron, for instance. How do they travel that watery system? Why I said that this can be a lesson for younger people? Because nobody asked for, after maybe 25 years of study of the endocannabinoids, how this happened. And instead, a young mind should pose this question maybe as the first or the second, because we know that endocannabinoids are lipids. They do have an hydrophilic tail, but they are lipids. They cannot move in water on their own. So it should have been obvious to immediately interrogate how this trafficking inside and outside the cell could occur. Instead, this is a very new point raised by a few groups, and I think it is an action point for developing new drugs that could be beneficial. So, trafficking, movement, how this really occurs with these compounds.
En este fragmento de la conversación, la doctora Cubillos plantea la importancia de entender, a nivel celular, el comportamiento de los fitocannabinoides y pregunta al doctor Maccarrone si existen otras áreas o cuestiones de investigación relevantes que deberían recibir más atención en el campo. El profesor Maccarrone responde que un tema crucial sin resolver es el transporte de endocannabinoides y fitocannabinoides a través de la membrana celular. Aunque hace años se identificó un posible transportador de anandamida en la membrana, su naturaleza sigue siendo incierta. No se sabe si realmente existe, si está presente en todas las células, si es una sola proteína o un conjunto de proteínas que trabajan en conjunto. Destaca que esclarecer este mecanismo es fundamental, ya que manipular el transporte podría tener efectos terapéuticos importantes. El Dr. Maccarrone subraya que el conocimiento actual sobre cómo los endocannabinoides se desplazan dentro y fuera de la célula, especialmente en entornos acuosos como la sinapsis, es muy limitado. Señala que, a pesar de décadas de estudio, preguntas básicas sobre este proceso siguen sin respuesta, y anima a las nuevas generaciones de científicos a investigar cómo estos compuestos, que son lípidos y no se mueven fácilmente en el agua, logran su tráfico celular. Considera que comprender este mecanismo es un punto clave para el desarrollo de nuevos fármacos beneficiosos.
Sounds like it's going to be a rich area for people to start asking the right questions and hopefully, as you mentioned, develop pharmaceutical agents that have an impact on these systems or these transport systems, and then can increase the availability of the endocannabinoids, or potentially work as well, or be manipulated to work with external cannabinoids as well. So,
Paola, sorry, let me elaborate a little bit more on this. We learned by experience that when you have a nice antagonist of a receptor or an inhibitor of an enzyme, you can have side effects that can be much worse than the beneficial effects. Now, let's imagine a cell with many targets for the endocannabinoids, but the cell having transporters that know where to carry the endocannabinoid. Usually, I like to say that when you arrive at the airport of a new unknown city, let's say that if you take the yellow taxi, you know that will go to the hotel. If you take the white taxi, it will bring you to the conference hall. And if you take the red taxi, you might go downtown for having free time. So it is the taxi that can make the difference. And endocannabinoids have different taxis in the same cell. And there is evidence showing that one taxi can bring you to one place, for instance, to nuclear receptors. Now, it would be wonderful that by inhibiting the right taxi, not the final destination, the entity, the receptor, the enzyme, but the taxi, you could block selectively all the activities of the endocannabinoid on the target, but only on that target. Okay? So this could potentially reduce the problem of side effects, unwanted effects, really a lot. You don't stop CB1 and all CB1 that you have in your body. You can stop the red taxi bringing that endocannabinoid to CB1 in that cell type. I'm really simplifying, hopefully not too much, but these molecules could be really a powerful point of action for new drugs, for thinking of new therapeutics.
Mauro Maccarrone explica que, a partir de la experiencia, se ha comprobado que el uso de antagonistas de receptores o inhibidores de enzimas puede acarrear efectos secundarios que superan los beneficios. Utiliza la metáfora de los taxis en un aeropuerto para ilustrar cómo, en una célula, existen diferentes taxis o transportadores para los endocannabinoides, cada uno llevando la molécula a un destino específico dentro de la célula, como los receptores nucleares. El profesor Maccarrone sugiere que, si se lograra inhibir selectivamente el transportador adecuado y no el destino final, como el receptor o la enzima, sería posible bloquear las acciones del endocannabinoide solamente en el objetivo deseado, minimizando así los efectos adversos. De este modo, no se bloquearía todo el receptor CB1 en el organismo, sino solo el transporte concreto hacia ese receptor en un tipo celular específico. Concluye que estas moléculas representan un punto de acción muy potente para el desarrollo de nuevos fármacos y terapias.
That is a really good point. And my understanding is that there are now pharmaceuticals being developed to try to target some of the compounds that are transporting things in and out, endocannabinoids, in and out of the cell as well. So sounds like a promising therapeutic avenue as well to explore. So we'll stay tuned for more developments in this area as more people focus on this. So just to cap off our conversation, Dr. Maccarrone, and this has been really, really enlightening, and I really appreciate you sharing so candidly as well and trying to explain and simplifying things for our audience. I know these concepts sometimes can be a little bit tricky, but I just wanted to ask, we've talked about what the field you think needs to focus on, the questions that are not being asked. What advances or what developments do you think will transform this field in the next 5 to 10 years? In addition to addressing the transport systems, is there any other area that you think is going to have a huge impact in the near future?
Yeah, well, again, it might seem strange, but out of the many proteins that are around an endocannabinoid signaling receptors, some of the transporters that have been identified, many metabolic enzymes that apparently are even redundant. You have 5 different ways to synthesize anandamide. We know the 3-dimensional structure of a few. So we need to do basic research, and we need scientists, young minds that like to do it, because without that knowledge, we can develop whatever molecule you like, and the chemist or synthetic chemist, organic chemist then build, but you will never know why there can be side effects. So we don't know the structures of too many elements of a classical endocannabinoid signal system. This is one. The second is As I mentioned, we are learning that there are different layers of complexity in the interactions of lipids with other lipids, endocannabinoids with eicosanoids, with specialized pro-resolving mediators, with sphingosine 1-phosphate. All these things are in a way a frontier of the field. And I think that if we take the right advantage of artificial intelligence, this complexity can be handled. I'm not saying that artificial intelligence will tell us, in much the same way as omic techniques have not given real answers. You have to ask the right question to get the right answer from a powerful methodology, right? But I'm sure that the complexity of data, the amount of data that you have to handle to try to see how these interactions and this orchestra of lipids can work, can be made possible by artificial intelligence and these new tools that we have in hand. So I hope that I can witness a quantum leap in our appreciation of this endocannabinoid signaling at large.
En respuesta a la pregunta sobre qué avances podrían transformar el campo en los próximos 5 a 10 años, el doctor Mauro Maccarrone destaca la importancia de profundizar en la investigación básica sobre el sistema endocannabinoide. Señala que, aunque existen muchas proteínas y receptores asociados, aún hay numerosos transportadores y enzimas metabólicas cuya función y estructura tridimensional no se comprenden completamente. Maccarrone subraya la necesidad de que jóvenes científicos se dediquen a este tipo de investigación para poder comprender los posibles efectos secundarios de nuevas moléculas antes de su desarrollo por los químicos. Además, menciona la creciente complejidad en las interacciones entre lípidos y otros mediadores, lo que representa una frontera en el campo. Finalmente, resalta el potencial de la inteligencia artificial y nuevas herramientas para manejar la enorme cantidad de datos y descifrar cómo funciona la orquesta de los lípidos, esperando presenciar un salto cualitativo en la comprensión del sistema de señalización endocannabinoide en el futuro cercano.
Excellent point about artificial intelligence too. It's definitely a tool that looks very promising and that it's going to help us make sense of so much information that is out there, but definitely requires skillful researchers, really grounded in methodology, really grounded in the proper techniques to be able to understand what the artificial intelligence output is and how useful it can be. So great points to look to the future as well, which leads me to my final question. There are many young researchers in Latin America that are hoping to make sort of strides and start sort of exploring in this field. What advice would you have for them other than focusing on the transport system and focusing on these areas that we just identified as promising areas? What advice would you have for them?
Well, the way a young scientist can grow up and express himself or himself can be not just one, but so many. So it's difficult to give a recipe for success, let's say. But I can think of myself and tell what happened to me. I anticipated it at the very beginning. I was very much interested in an enzyme, lipoxygenase, because of the properties of the enzyme. In those times, maybe someone that is a little bit older might even remember, there was a very promising new family of enzymes called kinoproteins with the potential of giving a Nobel Prize to this founder. And lipoxygenase seemed to be one of those. So my first years of PhD were devoted to show that unfortunately it was not. And then you see, you are a biochemist, protein biochemist, looking at the cofactors of an important enzyme. And one day you read in a catalog from Sigma that there is a new molecule that can be a substrate of this lipoxygenase. Now you have to be curious because that molecule is not exactly the same as the classical substrate. And you can wonder why nature has done something like this. Why this little piece of molecule is added to the natural substance. And step by step, collaborating with many colleagues, thinking about possible implications, you end up in working on a new class of lipids, the endocannabinoids, for 30 years. You see, next corner, there can be something that is not totally obvious, and the young mind can be attracted by that rather than by the routine or most popular, most fashionable trends of science of her or his times. So try to be curious but also original.
Frente a la pregunta sobre qué consejo le daría a jóvenes investigadores latinoamericanos que buscan entrar en este campo, el doctor Mauro Maccarrone comparte que no existe una única fórmula para el éxito en la carrera científica. Cada joven investigador puede encontrar su propio camino. Relata cómo su interés inicial por una enzima lo llevó a investigar nuevas familias de proteínas, y aunque sus primeros hallazgos no resultaron como esperaba, su curiosidad lo impulsó a seguir explorando. Gracias a la colaboración con colegas y a la apertura para cuestionar lo establecido, acabó dedicando 30 años al estudio de los endocannabinoides, una clase novedosa de lípidos. El profesor Maccarrone aconseja a los jóvenes científicos que cultiven la curiosidad y la originalidad, buscando oportunidades más allá de las tendencias populares y rutinarias de la ciencia, ya que en cada esquina puede surgir una nueva y fascinante dirección.
Curiosity, an open mind, perseverance is what I'm hearing in all of your advice. Yeah, these are really interesting times for science. I won't deny that there's so much going on that is going to affect the outlook, but definitely all the efforts that have been made by thousands of researchers around the world are there to make our lives better, to understand how things work in a better way so that human beings ultimately can make better decisions about, in this case, about our health. It's important to keep an open mind and to remain curious to little quirky things that one might find in one's field, and to surround yourself with the right people, with the right resources, and being able to reach out. I imagine reaching out, community building, collaborating with others is something that I've seen is a feature of this field, and I'm glad that that our researchers in Latin America could potentially tap into those resources as well. So with that, I have to finish this lovely conversation with you, Dr. Maccarrone. Thank you so very much for your time and your willingness to share a little bit of your journey. Always a pleasure to learn from you and to listen to you. And I cannot thank you enough for the work that you've done, for the contributions that you've made to this field. And I look forward to continuing learning from you and your incredible work. Y para quienes nos escuchan en Latinoamérica, muchas gracias nuevamente por escuchar este podcast. No duden en compartir, en comentar en las notas de este episodio. Nuevamente, mi nombre es Paola Cubillos, soy miembro fundador de la Sociedad Latinoamericana Interdisciplinaria de Cannabinología y con gusto para mí dirigir este episodio y nos vemos en el próximo episodio de este podcast. Muchas gracias.
Muchas gracias.